large protein. And you should want add some units of, for example, these are units of carbohydrates. Okay, exactly like a phosphate, we have a specific site for the binding of the oligosaccharide. For example, this is an oligosaccharide. Amide group of asparagine and hydroxyl group of serine and also threonine, they serve as a site of attachment. So, as I mentioned here, sometimes you have a serine and threonine, and sometimes you have it here. You have it asparagine. These are abbreviations, okay? So, for example, glucose, mannose, arabinose, okay? They want bind to this protein. This is your protein. So, totally it's considered as a glycoprotein, okay? Protein, oligosaccharide. Overall, we consider as a glycoprotein. But as I mentioned about the binding site of the oligosaccharide, only few amino acids they can bind. Asparagine, serine, and threonine. And we have the O-link glycoprotein. I don't know, Dr. Rebecca, will you explain or not? And N-link glycoprotein. Do you remember? For example, this is your protein. You have a one nitrogen as a bridge. And for example, these are carbohydrates. I don't know, glucose, mannose, and other type. And also you have another type. This is your protein. Oxygen as a bridge. For example, glucose, galactose, and etc. Okay? So as you can see here, we have two different types. O-linked glycoprotein, and what about this one? N-linked amino acid. Okay, but please tell me, I want to know, do you understand the difference between the different structure of amino acid or not? Whenever you have an O-link glycoprotein, which amino acid present here? O-link. Absolutely, you should have a hydroxyl here to provide the oxygen as a bridge. So, which amino acid can we sit here?
Serine.
Excellent. Serine and threonine. What about this one? Asparagine. Because asparagine itself contains a nitrogen, it's donates its hand to the, for example, glucose, to the galactose. Okay? So, generally, we have two types of glycoprotein. One of them is O-linked, another one is N-linked. So, if I ask you about the name of the amino acid, they provide a link between protein and carbohydrate part of your molecule. About O-link, this is amino acid serine and threonine, okay? And about the N-link, this is asparagine.
Doctor?
Yes?
I have a question. You say the amino acids have, you say the enzymes have 100 amino acids. That's
more than maybe 100,
yes.
1,000 amino acids.
That's made the structure of enzyme the same of amino acids?
You know, we have it, I will explain about how the 3D structure form. At first, amino acids bind together, they form a primary structure. then they form some helix, and then they form a 3D structure. I will explain, okay, in the next slide.
I mean, the protein and
You
enzymes
know-
have the same structure?
Yes.
Or not?
No. You know, what is the nature of enzyme? Enzyme is one of the example of protein. Okay?
Yes.
For example, antibody is one example of protein. I don't know. Actin, the contractile, yes, in the muscle is one of the example of protein. All of them are example of protein. Okay?
I understand. Thank you.
You're welcome. So, about this slide, what is important for your final exam? Actually, I never ask you some question about this slide. But I want to direct your attention to the difference name, okay? Because I want to understand. Who can tell me what is the difference between aspartic acid and aspartate? Aspartic acid and aspartate. As I mentioned in the previous slide, I asked you to memorize about the name of the negatively charged. So what is important in this slide? Negatively charged amino acids are aspartate and glutamate. But as you can see here, we have an aspartic acid, aspartate. What is the difference?
One is donor, one is acceptor.
Aspartic acid has a hydrogen atom. When it gets ionized, it releases the H proton. So it basically a proton donor. That's why it's called aspartic acid.
Excellent.
So when you release it, it becomes aspartate. When you lose the hydrogen atom attached to the COO. COO is negative, H is positive, so it becomes charged negatively.
Yes. Do you want to add more information? Okay. So as your friend mentioned and as you understand, whenever you want to emphasize that your molecule present with the negative charge, I mean it loses proton, it represents as an aspartate or glutamate. But whenever you use the aspartic acid, glutamic acid, it means that it takes the hydrogen, okay? And there is no net charge in the R group. About the details of actually is not important, okay? What is important here about this slide? I want to review for your final exam. At first, maybe I ask you about the name of the positively charged lysine, arginine, and histidine. Like tryptophan, arginine contain one group with a specific name. This is a guanidino. So if I ask you, guanidino containing amino acid is arginine. Or maybe I ask you, arginine containing indole, guanidino, I don't know, like some, for example, imidazole group or other name. This is a guanidino. And about histidine, histidine containing one specific group known as a imidazole. Okay? So we have a three names specific for our group. Tryptophan, indole, arginine, guanidinol, histidine, imidazole. Okay. Another point.
Doctor,
Yes.
what's the name of
The name. There is no specific name for the R group of lysine.
just last?
Just lysine. Just know that lysine is a basic amino acid or positively charged amino acid. Okay? So, as you know, whenever you have a positive charge, it means that, for example, lysine can release a proton. This means that this is a base molecule. Are you familiar with the function of acid of base? Acid. Acid can take or lose a proton.
Donate proton.
Okay, so base actually, but and it's better to say that, you know, especially for the histidine, generally it considered as a base molecule. It means that it can take a proton, okay? But in some condition, in some specific pH, histidine also can release a proton, okay? So we have an exception here, you should know that, especially regarding to the histidine. You know, in some pH and sometimes in a physiological pH, histidine considered as a specific buffer, okay? So if I ask you about the name of the amino acid that can function as a buffer in the protein, this is exception, this is histidine. And inshallah for the next session, I will talk about one of the role of the hemoglobin. Hemoglobin is one of the buffer molecule, okay? Why we say hemoglobin is buffer? Because it contains a specific histidine molecule. And because of the presence of histidine, we say that hemoglobin is buffer, okay? So, what is important in this slide? At first, you should know that they are basic amino acid, lysine, arginine, histidine. Another point important to memorize, the specific name of our group, as I mentioned, there is no specific name for our group of lysine, but about arginine, this is guanidinol, about histidine, this is imidazole. And another point is are some explanation. Just know that histidine considered as a bufferic amino acid. Okay? About the details for your level, there is no need. Just know that about the bufferic system, histidine provides a major role. This slide is for more information. As you can see here, we have a 3-letter abbreviation and 1-letter symbol for each amino acid. But for your level, there is no need. For example, as you can see here, for example, I want to represent serine. I didn't write the full name. I write there, for example, abbreviation. About the threonine, THR. About tyrosine, TYR. Okay, these are some abbreviation. But for your level, there is no need to memorize this slide. Okay, so it's better to put a red star. We can actually not red color here, but there is no need to memorize the abbreviation. Okay, but what is the importance of the nature of the amino acid? I kill myself to describe the different type of amino acid. But what is the importance? And what is the, yes?
A lot of genetic diseases, the reason for their happening is because of malfunction in the basic structure of amino acids.
Excellent.
Like for example,
sickle cell anemia.
Sickle cell anemia or maybe scurvy, but like it's not genetic.
Yes.
Yeah, and there is also another syndrome, I think. I forgot its name, but yeah, it's all about the amino acids. If there's a malfunction, the structure isn't normal, then a disease happens.
So, many thanks. So, as you can see here, we have one specific disorder now as a sickle cell anemia. This is a normal structure, and this is an abnormal structure or a mutant form of the protein. As you can see here, even one substitution of one amino acid, glutamic acid. What is the nature of this amino acid? Glutamic acid is acidic amino acid. It contains a negative charge here after replacement with valine. If you remember, valine is non-polar molecule. So you change a polar into the non-polar molecule. It can cause a genetic disorder, abnormality about the RBC. It's known as a sickle cell anemia. So as you can see here, there is a close relation between a structure and function. When a structure is normal, function is normal. But whenever the structure is abnormal, for example, they cannot transfer oxygen, so it results in an anemia. So maybe I ask you in your final exam about this keyword. The sickle cell anemia results from this part. What about the which subunit? There's no need to memorize. Just know that if I ask you what is the problem in sickle cell anemia, this is a replacement, okay, change of one amino acid with a negative charge with the non-polar amino acid, okay? And also, in addition to, as you can see here, it's contained in red stars, just for more information. As you know, we have a more than 20 amino acid. For example, these are some specific amino acid. Hydroxyproline, hydroxylysine. They derive from amino acid, and in some parts of the body, we have a different. For example, in a urea cycle, in a collagen, and other parts of the body, we have some derivatives of the amino acids. And according to the actually nutritional aspect, we have essential, semi-essential, and non-essential. There is no need to memorize, okay? Because this is controversial issue. And about the optical properties of amino acid, I want to ask a question. Which form of glucose is predominant? D-glucose or L-glucose? In the nature. About monosaccharide. Excellent, D form, okay? So, for monosaccharides, D form is important. But about the amino acid, which form is important? The L form, excellent. So, in the structure of protein, you have L-serine, L-glycine, L-tyrosine, L-threonine, okay? But we have another type, D-type, for example, D-serine. We have a D form of amino acid in antibiotics and bacterial cell wall, okay? Normally, in the structure of protein, they cannot participate, okay? But in, for example, antibiotics, in a bacterial cell wall, you can find this type. And how can classify according to the direction of amine group? Whenever amine group, I mean the free hand of the amine group, is located on the right, this is D form. Whenever it is left, this is not actually correct because I want to use some words to memorize it better, okay? Please pay attention. L is not originated from left, okay? I want to use, for example, whenever, for the first time, when I was a student, many years ago. When for the first time I studied this actually type of isomer, I recall that L left, okay? So the amine group, whenever located on the left, it's considered as a L. And as I mentioned, the L form is predominant, okay? L-serine, L-histidine, L-lysine. So they consider as an optical. Another point, here. And most of the time I ask you about this sentence. As I mentioned, glycine is not chiral. Why? Because
It doesn't have an R group.
it doesn't have 4 different groups because the R group is H. Okay? So if I ask you which amino acid is not chiral, this is glycine. Wallah, I review all of the sample question. Okay? If you make a note, you can actually form some sample question. And for a final exam, just review this sample. Okay? I take a note. All of them, actually, I mentioned all of the sample question. Okay? And about some properties of amino acid, you know, protein can be mutated. Okay? Following the exposure of the UV light. Why UV is dangerous? Because it can cause a mutation. Okay? It can cause a mutation. But which amino acid can absorb UV light? Excellent. The most important amino acid can absorb UV light. This is tryptophan. And to the lesser extent, tyrosine. As you can see here, both of them are aromatic amino acid. So, if I ask you about the, for example, the reason of the absorption of UV light is the presence of aromatic amino acid. But which of them is the most important? Tryptophan. And to the lesser extent, tyrosine can absorb UV light. So in this slide, you should memorize tyrosine, tryptophan, and UV light. I want to start a new topic, so it's better to have a rest time. Yes? Because it seems that you are tired. After 10 minutes, okay?